Welcome everybody back. It's a pleasure to introduce Adrienne Fairhall, who's a professor in the Physiology and Biophysics Department at the University of Washington, as you see, and she is, leading a lot of wonderful theoretical and computational neuroscience initiatives at UW and also elsewhere. Adrienne, just briefly, she started in physics, like many people in theoretical neuroscience, back in Australia already as an undergraduate, and got her PhD in Israel at the Weizmann Institute, also in theoretical physics, and then moved to the US to do, I don't know at what point exactly you transitioned to neuroscience, but somewhere between NEC and Princeton University, somewhere there in that area, she started working in theoretical computational neuroscience. Started a faculty position in Seattle in, I think, around 2003, 2004. And has been there, and she is probably one of the most accomplished and well-known theoretical computational neuroscientists. So it's a distinct pleasure to have you tell us a little bit about neural coding and adaptation today. Thanks, Adrienne.
Great, thanks, Venky, so much for the invitation to join you there. I would have loved to actually join you there. I've always wanted to visit India, and it would have been a great opportunity to do it. But nonetheless, it's great to have the chance to talk to you all. So I'm going to take off in a little bit of a different direction than the lectures you heard about this morning from Sahita. So there are a couple of different angles into neuroscience, and one of them that's super important is to really be able to write down in detail sort of the physics of the dynamical systems that underpin the brain. But today we're going to kind of zoom back a tiny bit and think about what those events in the brain actually mean and what they tell you about what's going on in the world, and hopefully eventually what they can tell you about computation in the brain. And so we're going to go through just the idea of neural coding. What does it mean when we talk about neural coding? Talk about some basic methods that we use to characterize coding. And hopefully for the last half, if all timing goes well, I'm going to tell you that, that we have this nice idea that neurons encode information, that is sort of a moving target. Neurons have a lot of intrinsic dynamics, as do neural circuits, that mean that the relationships between inputs and outputs vary over time. And so I'm going to go through a couple of different ways in which we see that unfold. And generally what turns out to be quite interesting is those relationships, and the way that coding changes through adaptation often has a very sort of satisfying mathematical description as well. And so one can see it as actually being good for coding, in some information theoretic sense. So hopefully we'll get to that. Great. So what even could we mean by neural coding? Let me just foreshadow that it is somewhat of a loaded term, and that when we're talking about a neuron encoding information, we have to see that in the context of all the complicated dynamics within the brain. So are you seeing also there, if I hide that, great. So right, neurons are connected together in a complex neural network with nonlinear interactions between them. Some fraction of them are getting inputs from somewhere else, but this network itself has intrinsic dynamics, as do all of the neurons themselves. There are inputs that we are not observing. So what are we trying to do in coding? We're trying to describe the relationship between, say, an input here and the firing rate, maybe of one neuron, maybe of a population of neurons. But we're doing that within the context that that there's all kinds of stuff you're not really capturing, right? There may be inputs that we don't see. There may be properties of modulation or top-down influences that are coming in that we're also not able to characterize. And so this relationship is always going to be approximate and somewhat, generally we can think about it as sort of probabilistic, because we're not taking into account many of those other factors. One might also argue that it may not be even correct to think about the brain as representing information. And there's some interesting recent papers that really sort of philosophically challenge that idea. I think you can go a long way with that idea. And in fact, I think we're a little bit lost without it. And so I think try to just bear with us throughout this talk to just see how far one can go by trying to understand these relationships. So in general, if we go back to this image, right, imagine we have all these neurons in, let's imagine we're, have a light sheet microscope and we're recording optically from some subset of neurons. We're able to track their firing rates. And now I'm going to plot those activities of those different neurons in some, here 3-dimensional, but ultimately n-dimensional space if we have n neurons. And those evolve over time. So what we're ever doing, right, is taking some snapshot of those firing rates at some time and trying to characterize what structure we're seeing there. So one could write down an equation for the behavior of that network in terms of the time, the temporal behavior of those firing rates. And so one very generic way to write that down would be that the firing rates are changing over time according to their current value, right? So their value at that same time, plus some set of inputs, and there's some nonlinear function f that acts on all of those things. This is just a very generic way of thinking about what a neural network does. And so there's lots of questions one could then ask. And the one we'll be focusing on is, what is the relationship between these parameters or observables are, our firing rates, and external variables in the world that we can observe or control. Hopefully, I imagine that you will be hearing more within this school about broader questions, or what can one infer about brain state from observations of R? Is R, this measurements of firing rates even the right variable set? What sort of low dimensional structure do these response states have? And finally, what dynamics does this network carry out, and what computation does it embody? And so I think answering some of these questions is really where you're going to get to over the next week in the course. But we'll kind of zoom in here. What's the relationship between external variables and firing rates? And so here, what you're looking at are responses from retinal ganglion cells. And so the retina is one place where the idea of coding has been pretty well fleshed out and works actually very well. So here, every one of, this is a raster plot, so some of you haven't seen this kind of representation of data before. What happened in this experiment was that you have a retina that's been taken out of the eye, it's been laid in a dish, and it's been shown movies over and over again, because the retina is sort of an outpost of the brain. It's basically a feed-forward network, and so we assume it's working outside the brain almost as it does when it's inside the in its natural biological context in the eye, because almost nothing is coming back from the brain. So if you play the same movie over and over again, these red dots show you where the times at which different neurons fire. And what I hope you can see from an image like this is that individual neurons are firing at very precisely controlled times. So this was a natural movie. I think it was Casablanca or something like that. And so say cell P, right, this neuron is firing precisely at very specific moments in the movie. There's a little bit of variability, but fires reliably and regularly at those times. Different neurons, some of them fire at the same event at the same time, some of them fire at completely different times. So each of them seems to have something, right, about the input that they specifically care about, and whenever that thing happens in the movie, they fire. And so under this, I think an image like this really does give you the sense that a code is a good way to think about the responses of neurons like this. So ultimately, what one wants to do is to be able to build a model that relates, say, input to output. In the, let's imagine we have a sequence of images in a movie. We're able to record the responses of many neurons. We would like to build a model that tells us what's the probability of seeing a certain neural response given a certain input. And ultimately, we may also want to turn that around and think about that in terms of predicting what the animal is going to do. So if we observe, now let's take X to be, say, behavior. Here, we'll talk about more about this tomorrow. This is an animal. This is Hydra, so like a 1 millimeter jellyfish, in which you can see all of the neurons in the animal responding. So we can get a complete picture of everything that the nervous system is doing. And one could similarly write down a model for what's the probability of a certain behavior given observations of the neural network. So why is that hard? It's hard, you can already see, from a cartoon like this, that if we really want to write down this as a probabilistic model, and here in Hydra, for example, we have 600 neurons, that's a very big probability distribution to build. If we wanted to think here in the retina about the responses, even of a single neuron to an image sequence like this, in order to capture a description of that image sequence, you need to tell me, right, the intensities of all of the pixels in an image over perhaps 100 frames. And so that is a description, the size of that piece of information, right? What happened in the movie at a moment prior to this cell firing is thousands of numbers, right? So to build this model, to build this distribution is a lot of numbers. And if we had to really fill up the entire million-dimensional distribution, we would just never do it. We'd never have enough data. And so ultimately what one would like to do is to be able to measure properties of the environment. This might be the intensity of pixel 1, intensity of pixel 2, or it might be different properties of the inputs, such as concentration of an olfactant or sound wave, the pressure in a sound wave. So we'd like to take all of that information and kind of crunch it down to some smaller subset of features of the environment that can be captured by some kind of low dimensional model, and do the same ultimately for neural activity and do the same ultimately for behavior, and to build a model that relates these lower dimensional descriptions to one another. So our goal today, and this, we'll come back to this tomorrow. Normally we think about relationships between environment, neural activity, and behavior. Ultimately, one might also like to incorporate the muscles in that story. And tomorrow I'll tell you a bit about work that we've done that actually incorporates the muscle activations into a model and allows us to take this picture one step further. Yeah, so just to emphasize, though, that even when we've done this, right, if we did manage to find this grand model whereby we could understand low dimensional descriptions of the environment, the neurons, even the muscles and the behavior, that is still just a statistical picture of what's going on. And it still doesn't, unless we apply some kind of insight to it, doesn't necessarily reveal the algorithm that's underlying the whole thing. And so while a lot of sensory neuroscience has focused really in the last 100 years on trying to determine what is it that this neuron cares about or is responding to, it's really just the beginning of what we really think about as the goal of computational neuroscience, which is to reveal the underlying algorithms. And so it's always, I think, going to be a bit of a back and forth between, sophisticated, potentially machine learning ideas about how to extract information about what spikes are telling us, and then interpreting them in terms of some actual computation or algorithm. All right, so as I kind of referred to already, what we mean by neural coding is fundamentally a quest to reduce the dimensionality of this problem of finding relationships between neural activity and sensory inputs or behavioral outputs. And so, ideally one would like to be able to just figure out, right, what is the stimulus parameter of interest to a given neuron, and then to map out the neural response. So here would be just a very simple cartoon of a tuning curve, right? How is this neuron and its firing rate tuned to some aspect of the stimulus? So it's ultimately, ideally where we would end up. We would have very complicated set of recordings from the brain. We would be able to stimulate our neuron with a rich set of inputs, but ultimately we'd like to, in order for us to really to understand what's going on, we need to be able to simplify it, perhaps not to something quite this simple, but something that's sort of comprehensible. All right, so let's just talk briefly about what we mean by dimensionality reduction. I don't know how interactive we can be here, but I'd kind of like to see, I'm going to pull up the chat, kind of like to see what people know already about dimensionality reduction. So when we use that term, we mean that we have some, here I'd like to think about something that's varying in time, that we would like to be able to express it as a set of basis functions where they may have time-varying coefficients. And these basis functions could be themselves sort of high-dimensional objects, but now if we are able to re-express our input or our, let's say, our movie in terms of a set of basis functions multiplied by these coefficients, the hope is that we would have fewer coefficients than, say, pixels in the movie, right? So anyone want to throw out into the chat? I opened up the chat. Some methods that you already know about that would allow you to do something like this. PCA, great. Yes, PCA. Okay, we got PCA, which is a very fundamental one. Anyone know any maybe even more simple ones? T-SNE, excellent. Yes, Fourier transform, fantastic. Getting almost everything that I was looking for. Mode decomposition, yep. All right, let me take away the box. Yep, I think you've done well in picking up almost everything I was hoping that you would see. Very good. Manifold methods, wonderful. Yeah, so Fourier analysis is one very straightforward example of that, right? If you have some time varying signal, one might like to write it in terms of a decomposition into different frequencies, and now you just need to tell me what is the amplitude and phase of those different frequencies, and I don't have to tell you what the value of the sine wave is at every moment in time. PCA is another great example. There are variants, PCA-like ideas that instead focus on trying to make, to look at the structure of the data and to find descriptions of that data that are sparse, right? That concentrate the power onto as few modes as possible. So sparse coding, and some examples of that are independent component analysis. Someone mentioned T-SNE. So these methods are all linear. Fourier analysis and PCA is linear. Other methods are nonlinear. T-SNE is an example of a nonlinear method that tries to find an embedding that then takes all the data and kind of splatters it down into a low dimensional space according to what can be a nonlinear measure of the distance between the points. Another method that is becoming increasingly popular is variational autoencoders. So that is very nonlinear method that uses a neural network to take in the data, find some low dimensional feature basis, and then use that, optimize it in order to reconstruct your original data using as few modes kind of in this bottleneck as possible. Great, so people already have a reasonable sense of what we mean by dimensionality reduction. So let's see how that kind of plays out with neural coding. Right, so what we're going to focus on really for most of this talk is thinking about the firing rate of a single neuron and how that relates to properties of the input. All right, so we're just focusing on one of these guys. And now just sort of go from first principles to think about what kinds of models can be effective for understanding coding. Okay, so the most simple-minded thing that one could do, and a good starting point, because it's very helpful in sort of building intuition for more complex models, is to start with the assumption or that at least some aspect of this coding problem is linear. So we want to predict the firing rate at some time from a time varying input. Then a hypothesis is that what the neuron is doing is looking for some variation, some kind of feature within that input. And depending on how strongly represented that feature is in the input, then the firing rate is proportional to the strength of that feature. Right? So that sort of makes sense as an idea. How does that play out mathematically? Well, all that says is that we're going to take our responses at time t, and they're going to, let's just assume initially that they're just a linear function of the input, but not just of the input at one time, the input at a range of previous times. And so we take those inputs, looking backward from time t, we're going to step back in time by tau, so and then scan back over a whole range of taus. And at each distance backward in time tau, we're going to weight the input by a value by f, which is a function of tau. Right, so that is equivalent to taking our stimulus and projecting it basically onto that feature, and then assuming that our firing rate is just a linear function of the strength of that feature. Okay, so that tells us that these weights, if this, to the extent that this model is reasonable, these weights are kind of acting as a linear filter. Right? So this formulation that I've written down here, you might recognize from calculus is a convolution, right? So it takes some input and it convolves it with this set of weights f. So that's the same as filtering, which is the same as convolution. We can also think about it as projection in some high-dimensional space. So if we have our stimuli, which are defined by their sequential values in time, and we write we put down some axes such that every moment in time in our stimulus is defined by a different axis, then some random chunk of stimulus is going to be a point in this high-dimensional space. And so we imagine that our neuron is selective to only some subset of all of those stimuli, and let's hope that there's some structure to the stimuli that it responds to. And so this blob here, we're going to, in this example, are the spike triggering stimuli. And so one way to characterize that blob of spike triggering stimuli, a simple way to characterize them is just by their mean. And so this vector f here is the mean of that cloud, and that is a reasonable guess at what this feature is, right? So if we want to describe which stimuli are in that cloud, we can project that vector, say a vector here onto that feature, and that will tell us whether or not this is a spike triggering stimulus. And so if you have a different stimulus that's say over here, the projection onto that feature is 0, and so that is not in the cloud. All right, so that gives us a method, right, to determine sort of who's in this cloud. It needs to be oriented in the same direction as this feature f. It's not enough, right? That's not enough to characterize who's here. There's a piece that's missing, which is that of course, it isn't linear, right? The who's in the cloud is not defined simply by its projection onto F, although that does take us a long way in identifying where that cloud is. There's also a nonlinearity that says you don't fire, right? So if the projection onto F is small, this neuron doesn't spike. It will only fire when the stimulus gets large enough, right, to be out here in the cloud. So we need a second step in our model. And that is to say that not just the input should resemble this feature, it needs to be large enough. And one way to account for that in this example is that here as a function of the stimulus projected onto that feature, we can add in an additional step, a nonlinear function that tells us that the neuron is only going to fire when that projection is large enough, right? And so here, this is a kind of classic coding model in neuroscience. It's called the linear nonlinear model for good reasons, right? The first piece here is the identification of a linear feature in the input that's important to this neuron. And the second component is this decision function that says, Once I've identified that feature, I'm going to look at the strength of that feature that occurred in the input. And now there's some nonlinear function that relates the strength to the actual firing rate of the neuron. Right, so we can write that down in this form. So our response is some nonlinear function G acting on the stimulus projected onto that feature. If I feel like I'm always, what I hate about Zoom is not being able to see you or take questions kind of in real time. So since I have the chat not on screen right now, I'd be totally happy if people break in with questions, or if there are points of clarification that come up, maybe someone could, Venki or someone else could.
So in fact, there's a time that I've been sort of answering a couple of explanatory things, but there's one question just now about the interval of integration. And,
Right.
I just wrote it sort of 0 to some guess of a pass, so maybe you can elaborate on that.
Yeah, that's a great question. Here, let me stop and pull up the chat. Yeah, so in general, one would like, how far back in time, right? So what is the real meaning of that interval of integration, and what's the relevant, time scale? The real question is, how long back in time does a stimulus influence the spiking of a neuron now, right? And so that can differ for different systems. So yeah, in math, right, it just is 0 to some time t. In practice, how far back should you be collecting, right, your stimulus in order to predict the output? Just to give you a sense, in some real systems, in retinal ganglion cells, you need to look back maybe a couple of 100 milliseconds. There are some types of neurons in the auditory system that are super precise and very time locked. And so it's really only inputs about 10 milliseconds backward that influence them. As we'll talk about later, there are all kinds of longer timescale processes that can influence the way that neurons fire over even seconds. And so there is sort of a cascading series of influences. Typically, it's a couple of 100 milliseconds, but there can be long range influences that, if one were really building a complete model that deals with what we'll talk about later, deals with adaptation, then you may want to retain information over quite a bit longer than that. Okay. All right, so that is, kind of a cartoon picture, right? So there's a feature in the input that causes a spike. In general, that is a limited idea, because in general, one might imagine that there are multiple influences from the environment that are changing the probability of firing of this neuron. And one way to incorporate that is to simply, propose that there may be multiple features whose strength in the input combined together in potentially a nonlinear way to determine the output of the neuron. And so we can extend this model to think about multiple linear features. So the response at some time will be a nonlinear function of a combination of multiple features. And so what is helpful about a model like this is it, whereas in the olden days, one might have just done as Hubel and Wiesel and other very eminent neuroscientists who won Nobel Prizes did, they identified some feature of the input as being important. And in the case of Hubel and Wiesel, that was the orientation of a bar being relevant for visual neurons in the visual system in V1. And so then as you change the orientation of a grating or of a bar, you can kind of map out the firing rates of neurons to that bar, classic tuning curve idea. This more general idea, general picture, means that you don't necessarily have to specify in advance what that feature is. As I'll show you in a moment, we can use data analysis to determine what those features are, and then also to tell us what the kind of tuning curve relative to those features is. So it generalizes the idea of representation to any arbitrary stimulus feature that we can learn from the data, and any arbitrary nonlinearity. And so if we have a model like this that lets us quantify what neural representations are, how they change during adaptation and during learning, and even during development.
So Adrienne, there's a couple of questions that might be worth answering. So there's people asking, what do you mean by a feature? And also the question about like, can we give a physical example of sort of an unobserved input for
Right,
something?
right. Yeah, great. So let's, what I mean by a feature is so you might have had the word receptive field, right? That some neurons respond to something in their environment. And so here I'm really just thinking about, my drawing, right, is of a stimulus that varies in time. So let's imagine we're thinking about the retina, and this is the light level. Let's say we have the screen and it's flickering, and that's, there is some pattern of flickering, right, that causes a neuron to fire. It turns out that it needs to flicker in a certain over a certain trajectory, right, in a certain way in order for a certain neuron to fire. And that's what we're trying to identify in terms of this feature. So it's some property of the input. Here, we're just thinking about time, but that could also have been a spatial feature, and that's maybe more typical when we think about the retina, is that there's some spatial pattern, right, in an image that causes a neuron to fire. Let me see if I can even draw. I don't think I can annotate, can I? No. So your retinal ganglion cells are famously sensitive to a bright spot in the middle of a darker field. And so if that event, right, happens in the input at a certain time, then then a neuron, the retinal ganglion cell has a high probability of firing a spike. In general, one should really think about a spatiotemporal feature, that both space and time, that blob in the receptive field of the neuron has to kind of increase over time, and at that moment in time, that is going to trigger the neuron to fire. So, is that answering the question?
It seems reasonable. I mean, I guess bottom line is any sort of parameter of the stimulus, right? It could be color, or in the case sound intensity or sound frequency. This can all be features.
They can, right? But yeah, so yeah, let's go back a little bit here. So they can be features, but we want to be a little more detailed in what we mean by a feature, right? So S of T, as the example we just said, could be the intensity of a pixel or a region of space in the image. It could be the color of that region of space. It could be a sound wave. But here, what we're doing is building a model that takes it a bit further than just saying this neuron is sensitive to color, right? If S of T were red, right, that's fine. But in fact, what matters is that red, right, appears in the image with a certain temporal structure, right? It has to kind of, in this case of this neuron, if red turns on, right, if it changes in strength, then that's something that drives the neuron. So you should interpret this feature as a temporal variation in that component in the input that changes over time, and so that feature happened, and the neuron will fire here at time 0, right, in this picture. And so in order to find out when the neuron fires, what we're going to do is, now we know this feature. We know that this neuron fires when light kind of briefly, goes off and on again. Now we're going to look for moments in time when something like that occurs in the input. And how do we find those moments in time? Well, we take this feature and we project it, right? We filter the stimulus with that feature. So we look, when does this, say random sequence look like this? So we take this feature and we take the dot product of chunks of that stimulus S of T with this feature. And when that dot product is large, then that feature is strongly represented in the input. So similarly, if this were a spatial feature, we would take our movie and we would have, we would take our spatial filter and we would run it over the movie and say, when I look at this moment, this point in the movie, then that feature that my neuron cares about is strongly represented, right, in that image, and that will be the moment at which that neuron fires.
And to complete, Adrienne, the decision function is there for what? Because there was a question about why is that there?
Right. So the decision function is needed because, well, neurons, for example, can't fire negatively, right? So if this feature, if you had a moment in the stimulus in which exactly the opposite of this happened, right? There was a dip, then our linear model would just say that the neuron would fire negatively, and it can't do that. It also can't fire arbitrarily, at arbitrarily high firing rates. And so in principle, right, if there's something that neuron likes, we could just keep cranking up the amplitude of that feature, and a simple linear model would just say that the firing rate would just keep increasing indefinitely. But that's not the case. So what incorporating this decision function does is say, well, this deals with the fact that if the feature isn't there strongly enough, the cell just doesn't fire at all. But as I increase the strength of the feature, the firing rate will go up until some upper bound, and then it just can't fire anymore. And so it will just reach that, say 100 hertz, and can't fire more than that. And so this decision function just gives us a lot more freedom in translating from the existence of a feature that the neuron cares about in the input to the actual firing rate, which is limited by physiology. Right? It's rectified, right? It can't go below zero. It's bounded from above, can't fire arbitrarily fast, because there's a refractory period, as you heard about from Zahita. So that this extra piece lets us make a much more accurate model. That said, that a decision function like this is the most simple form of decision function that we know we need, right? We need to stop the neuron from firing, our model from having a negative firing rate. We need to stop it from going too high. But this is not the only decision function that one can generate. And we will talk a bit about that in a moment. So, I'm actually happy to take questions, because this is important to, at least, the core of this is important to all that we're going to go to. So this is a good point to be asking questions. All right. So now, when we have these multiple features, so let's imagine we have a cell that, so here's another example of a cell, and one you'll meet again, hopefully later on, is there's in the fly visual system, there's a neuron called H1, which responds to wide field horizontal motion, right? So when the scene that the fly is looking at moves in the horizontal direction, that neuron responds. But it responds to horizontal motion over a particular period of time, with a certain time scale. And so its feature would look like sort of an integration, a decaying integration of recent the recent velocity of that input. But it also responds a little bit to the acceleration, right? So it cares about velocity, and it also cares about acceleration. And so there would be a second feature that's the derivative of the first feature that also feeds into this decision function. So that you might, in general, if you're a motion sensitive neuron, you might care about motion and acceleration, and maybe even the third order derivative. Okay, so how in general do we figure out what these features are? And one very powerful approach to that is white noise analysis. So we take a random stimulus and we drive the neuron with this random stimulus, and we just try to build as big a data set as possible of stimuli and spikes. And so, so here's an example of that you might use to test a retinal ganglion cell. So what you're seeing here is a time-varying stimulus, where maybe you have an image that's divided into checkerboards. Every checkerboard has an intensity. And now every stripe here is sort of an unwinding, right, of that image into all the pixels. Maybe we're going to write it as one long vector, and that changes over time. So at this one column here is a set of pixel intensities, and then every frame is a unique draw of a different example of a random checkerboard. So we're going to just keep drawing those at random over and over again. So here, as we scroll through time, And then the neuron fires at particular times. So then what one should do is look back in time to say, okay, what were the specific examples of inputs that generated spikes in this neuron? So going back to this high dimensional picture, every one of these examples, right, of a time varying changes in pixel intensity is a point in some high dimensional space, right? We can take this chunk of stimuli. It's just a whole bunch of numbers, right, of variations over time and variations over space. There's a value of the intensity at every one of those coordinates, and that is some point in this high dimensional space. As we talked about before, maybe the simplest thing to do with all of those examples of spike triggering stimuli is to just take their mean. And so the mean gives us an estimate of the filter or the feature that the neuron cares about. And in order to calculate this nonlinear decision function, what we can then do is, let's remember what our goal was. We were trying to find out what the probability of seeing a spike is given the stimulus. If we were to try to figure out that model from this data, right? Stimulus here is defined by, let's imagine 6 time bins times, 8 pixels. That's a lot of numbers, right? That's a 48-dimensional distribution, and we will never fill up, the 48-dimensional distribution. And so what we'd like to do is to simplify the description of those examples, right, of these pieces of these chunks of stimulus that triggered the spike in terms of just 1 or 2 numbers. And the most convenient number to use is just how similar are they to the average stimulus. So if we stack them all on top of each other, we average them, that will give us the feature. And now we can ask how similar are any of these examples to that particular feature. Right, so here, here again, I'm just drawing it in time. The example we just saw also has a spatial component. So imagine that there's a spatial part here as well. So now in order to, instead of writing probability of spike given the entire stimulus, right, given all those 48 numbers, we're going to now think about the stimulus as being simplified. What we can do is use Bayes' rule to figure out how to write down that function from our data. And Bayes' rule tells us that we can rewrite this conditional distribution in terms of the probability of the stimulus given the spike, multiplied by the probability of the spike, divided by the probability of the stimulus. So we've just rearranged these conditional distributions. Probability of spike given stimulus times probability of stimulus is equal by laws of conditional probability to the probability of stimulus given spike by times the probability of the spike. Now, all the things on this side of the equation are things that we know. The probability of a spike is just the firing rate. We can just see how many spikes there were, and that will tell us the probability of seeing the spike. The probability of the stimulus given the spike we can get from the data that we saw before, right? We had a whole bunch of examples of that, so we can figure out what was the probability of the specific stimuli that triggered the spike. And the probability of the stimulus is something that we control ourselves. So we made the experiment, we generate stimuli with a particular distribution, so we know what that is. So everything on this side of the equation is measurable or known for our data, and that gives us sort of the answer of what this nonlinear function is. So just, let's just go through that again. So now, instead of thinking about our stimulus as this high dimensional thing, we're going to reduce it to 1 number for every spike, which is the stimulus convolved with the feature, right? So we take our stimulus, we project it onto the feature, in this case, just the mean. And now this input-output relation or nonlinear decision function just depends on this one parameter, which is S, which is the strength of the feature. And so we can gather from our data examples of what was the value of S at the times of the spike, and we can make that distribution. Probability of S is the thing we created, right? We generated a whole bunch of random numbers. Let's say they were Gaussian, And so the probability of the original stimulus is a Gaussian distribution. The probability of the stimuli given the spike is the thing we're measuring. So when has this worked well? This has worked well when the feature that we've found really does change the probability of firing. So if we were to look at the values of the stimulus, just randomly in time, because we made a Gaussian stimulus, that will just form a Gaussian distribution. We want that the moments in time at which there was a spike, as determined by our putative feature here, F, we want that to be different than the background distribution, right? So if we look at the probability of the values of the stimulus, given that there was a spike, and that just comes out to be the same as the background, then the spikes, they're not, the relationship is not characterized, right, by that choice of F. Right, so if you have a probability of stimulus given spike that is just a recapitulation, right, of the background distribution, these two distributions are the same, and our nonlinear function is not modulated by the value of S as determined by this feature. Whereas if that distribution is different, right, that the probability of stimuli at the times of spikes is different than the background, then we divide these two distributions by each other and we get something that's modulated by S, right? So for small values, none of the values of the background in this range triggered a spike. Only when they were larger do you start to see spiking, and that nonlinear function will go up. Okay, is that just checking on the questions, right? Yeah, so why is there some slope here? And that will generally happen, right? It won't be a super sharp transition from spiking to no spiking. Okay, so that tells you, what we talked about here is sort of a first pass at figuring out how to describe, right, this set of stimuli that trigger spikes, and that is through the mean of that blob. In general, even just looking at that blob, you might ask yourself, why do I stop there? I would get more information if I tried to characterize that blob by capturing more of its structure. And that is a good idea in general. One does do better by going to the next order moment, right? The lowest order moment of the distribution of this set of stimuli is just its mean. But why not also calculate its covariance, right? That characterizes kind of the shape of that blob in this high dimensional stimulus space. And many of you mentioned PCA before. You should hopefully recognize that that's exactly what PCA is trying to do, right? It tries to characterize the directions in the high dimensional space here that blob is occupying. So if one calculates the covariance of the spike triggering stimuli, or the points, the examples of the stimuli in this blob that causes a spike, and finds their eigenmodes, as one does with PCA, that gives you additional features that may be of interest, that are likely to be relevant to triggering the firing. Okay, so what do we mean by the covariance matrix of the spike conditional stimuli? So what we're going to do is take this time-varying stimulus at different times t, right? So here again, let's just go back to this one-dimensional stimulus. So light intensity, for example, so it's varying in time. Here are the moments of the spikes. What we're interested in is taking these chunks of stimulus as a vector. And now we're going to, starting at time t of the time of the spike, we're going to scroll back by I time points and subtract from that the spike triggered average, right? Subtract from that the mean of all of these examples. And then we'll multiply that by the same thing, right? But now we're going to take the jth time point back, right? So we're computing how different moments in time in this little piece of stimulus kind of co-vary with one another, right? So we take this vector, its value at time point I, we take the vector's value at time point j, and we multiply those by each other, and then we take the average. And so now this defines a matrix defined by time point I and time point j. And that matrix, its eigenmodes give us a basis for describing these chunks of stimulus. So again, remembering back to your intuitions about PCA, what we've done is to treat all of these temporal chunks of stimulus as vectors. They form some cloud, right, in a high dimensional space. By calculating the covariance matrix and its eigenbasis, we've found the directions of maximal variation in that cloud. All right, so here's what that will look like in general. So here is an example from similar to the picture that I showed you before, but now instead of looking in 3D, let's just look at it in 2D. So now we splatter all the non-spike triggering stimulus chunks, right? We can take this and just grab, random pieces of that stimulus and project it now onto the leading two eigenmodes of the covariance matrix. So we take this covariance matrix and we diagonalize it. We get back from that a series of vectors that tell us about the directions of structure, right, in the spike triggering stimulus space. And now when we look at all of the stimulus pieces in that high dimensional space, we see kind of a Gaussian blob in the middle. That's all of the stimuli that don't trigger spikes. And then the ones that do trigger spikes are different, right? They're oriented away from the origin generally, right? This purple vector is the mean of all of the spike triggering stimuli. And now the eigenvectors, the S1 and S2 tell us about the directions of structure of that blob. Okay, so how does this work when we apply it to some simple models of how neurons respond? And so there was some very nice work done by Marcus Meister's group years ago, where he with Pam Reinagel and Justin Keat wrote down a model that tried to capture, so compared to the sorts of models that you learned about this morning, where you would write down all of the ion channel dynamics in a single neuron and how that responds, say, to a current input. Here, what these guys tried to do, and it's a very important kind of line of computational neuroscience in general, they tried to write down a generative model for how a particular retinal ganglion cell responds to an input. And so if you know the input, then you can use this model to predict the times of spikes. And so in this case, the prediction was really exactly, the model instantiated a model of the type that we've been talking about already. So they assumed that the stimulus, S of t, is filtered through some feature, and that there's a nonlinear function applied to the output, right? So stimulus is convolved with that feature. And now once you have projected the stimulus onto that feature, there's a nonlinear function that tells you the times of spikes. And they took a simpler one. They just said that whenever that value, right, the convolution of the stimulus with the feature crosses some threshold, we're going to assume that that's the time that the spike occurs. Right, so here G of t, this is the output of the stimulus convolved with the feature. And here, every time it reaches threshold, you lay down a spike, and that's the estimate for how this retinal ganglion cell works. So you can probably tell that isn't great, right? That might give you some guess at when neurons fire, but really you're going to get a better model if you incorporate an additional feature. And so here they added to that model a kind of after spike potential. And so, generally speaking, the neuron won't just fire once, right? When you have a threshold crossing, it will continue to fire for some time. And so how do you capture that? What you can do is after the time of the spike, you can now generate what you could call an after spike potential that you add back into this generating function g of t. And that will, so let's watch what's going on here. So with, as we have an input coming in that drives this function g to go up, there's a spike. If we don't do anything, right, that value will continue, will just keep being driven by the input. But now we assume that there is some kind of internal dynamics of the neuron adds back into that potential function, some negative pulse, right? So that takes you back down again. And in this case, the input is not quite enough to counteract whatever that internal mechanism is in order to drive you above threshold again. So you stay below threshold and you stop firing. But this time here, now the input drives you up, you fire a spike, you generate this potential. It says there's something internal to the neuron, a refractory period that is going to stop the neuron from firing immediately. You add that in here and it takes you below threshold. But there's enough strength of the input still coming in that the sum of what you're getting from the input and that post-spike function now temporarily goes below threshold, but then comes back up again. And it will do that repeatedly because that whatever was happening in the input is sort of still strongly driving the neuron. Right. So now you get this sequence of spikes. And then eventually the input, dies away and you drop again below threshold. Okay, does that make sense? So this was a kind of constructed model, right? They assumed there was some feature. They assumed that there is some spike, internal neural dynamics that takes you away from threshold. And then they fit these two components of the model and were able to predict sequences of firing in neurons. So now we build that model and we use it as a model for what a retinal ganglion cell is doing. How does our method, right? How does our white noise analysis do in capturing the computation of this neuron? So what we would hope is that it reveals for us, right, whatever that feature was that drives the cell. So that would be neat if it did, but you probably are realizing that can't be the full story, right? We would hope that when we drive it with random inputs and we calculate the mean, that we will get back an estimate, right, of this feature that's driving the cell, because that's what it's pulling out of the input. But it's important to realize that all of this stuff also matters, right? The fact that the neuron only fires when it crosses threshold, and that now there's some internal mechanism that stops it firing, and then will only allow it to fire with some time delay, influences our estimate of what it is about the stimulus that's driving the cell. So in particular, what is always going to be true is that this neuron only fires when it's kind of on a rising phase, right? When that input is increasing in time, right? So when the stimulus convolved with the feature, when that value is increasing over time, those are the only times generally, or at least those are going to dominate the times at which the neuron fires. So what one might expect to see when you calculate this higher dimensional feature space is that it won't just be f of t that's important to driving the neuron. It will also be the time derivative of f of t. So one would predict that, in fact, if you calculate the space of features that drive the cell, you will see an influence, right, of the true feature that the neuron is pulling out of the input, but also of its time derivative, because that is going to strongly determine exactly what time the neuron fires. And that is what happens, right? If you take a model like this and you calculate, you take all the examples of stimuli that drive the spike, and then you calculate the covariance matrix and you pull out and you diagonalize it, you get two leading eigenmodes, right? So there are two features that are important for driving the cell, and then a whole bunch of background ones. So that's just sort of noise or features that don't matter. And what they look like is there's one that is exactly the feature, f of t, that drives the cell. And the other is its time derivative. And the spike triggered average here in black there isn't actually, it doesn't turn out to be f of t, right? So the spike triggered average is going to be a combination of f of t and the time derivative of f of t. And when you do covariance analysis, you really see that. You find that there's, in black, I don't think I've overlaid here what the original feature was, but in blue, that was the feature that we chose to drive the cell. And in red is the time derivative of the blue feature. And in black is the spike triggered average, which is now a combination of those two things. So now let's look at what stimuli look like when we project them into the space of these two features. Right, so here we've got three different clouds for three different examples of where we put the threshold. So remembering back here, we can keep increasing, right? We can choose some different examples of that threshold. And what's going to happen as we increase the values of those thresholds is that the importance of the original feature of f of t, right, that drives the cell, that shouldn't change. But what would change is the criterion for it to be only happening on the upward rising as that feature gets more important in the input, as that feature increases in the input. And so now these two dimensions of the stimulus that are being pulled out are feature one and feature two. So feature one is the true feature of the input. That's what came out of our covariance analysis. And feature two is the time derivative. And so here, what you see is that the projection onto feature one, as we keep pushing up the threshold, the values of feature one, right, because we've increased the threshold, they will increase with as we push up the threshold, because we've forced that to be true, right? So in the blue blob, that's when the threshold is quite low, so that feature doesn't have to be too big, right? But as we push up the threshold, we're going to push that blob across. Whereas, sorry, I misspoke before. So whereas the threshold mode, right, the requirement for that the neuron only fires as that stimulus is getting larger, that doesn't change, right? So projection onto feature two is the same in these three different cases, whereas the projection onto feature one gets pushed to higher and higher values. Okay, any questions
Adrienne,
about that?
can I interrupt maybe a couple of questions that might be timely to answer if you don't mind?
Definitely.
One is, what's the mechanistic explanation for the time derivative, why it's also a feature? And two, this kind of modeling seems good for externally driven stimuli. Like, what about neurons that code for some internal thing? Like, how do you deal with sort of generating white noise stimuli for those?
Right. Yeah, no, those are both great questions. So here, the mechanistic reason for the time derivative part is that is really because when we made our original model, it would work very well for a case in which you have a neuron that's just being presented with purely random stimuli, right? So in every time bin, it would be great, right, if the entire stimulus over the last 200 milliseconds had been newly, freshly generated, because then we would just ask in every time bin, are you or are you not like the previous feature? Real life doesn't happen like that. Real life scrolls in time, right? So there's a feature in the input that gets stronger over time as time progresses, right? So if we now plot here, what we're seeing in gray is is the stimulus being, as we scroll the filter right over the stimulus. And so that has temporal structure. And so what is the moment at which the neuron responds to that stimulus? Presumably it's the moment at which it first gets big enough, right, to drive the stimulus. And so that means that although at all these subsequent times, it's also big enough to drive the stimulus, it had a bias, right, to firing at the first time that it was big enough to drive the stimulus. So that says that there's going to be a contribution from the fact that the filtered stimulus is increasing in strength at the moment that the neuron fires, right? So that says that the time derivative of that input also has to be relevant, because most of these spikes are going to happen primarily at the first moment in time at which the stimulus crossed, the filtered stimulus crossed threshold. And then the second question was, what about, this is all very well for external stimuli. How does one ever think about what's going on internally when you're not controlling the stimulus? And that's a big open problem. And I'll say a little bit more about specific examples of that later, of how these methods can fail in some sense. All right, so just wanted to show you that, if you go through this with the retina, we do, while these guys built this beautiful kind of generative model for retinal ganglion cell firing, many retinal cells don't actually do that. So some of them, that's a great model. Some of them do have these two features, two relevant modes, one of which is a feature, one of which is its time derivative. They have, the blob of spike triggering stimuli looks just like what you would expect from this filter and fire model. But there are a lot of other examples that don't look like that at all. And so here you get multiple ligand modes. So, we didn't go through this. It really would, typically take a lot longer to lay out all the details of this. So here, the eigenvalue tells you something about the spread of the stimuli, the spike triggering stimuli with respect to that feature. And so a negative eigenmode says that the cloud of spike triggering stimuli is sort of smaller than the background distribution, whereas a positive eigenmode says that the spread is larger than the background distribution. And so with a filter and fire model, you always expect that the spike triggering distribution is shrunken relative to the background, right? So all the background stimuli were some Gaussian in the middle. The spike triggering stimuli are a restricted set of them, right, over this sort of narrow range, because they have to cross threshold in this dimension, right, to fire. And so you expect that variance, right, the spread of that distribution is smaller. There were cases, however, in which the distribution of spike triggering stimuli relative to some feature in the input was actually increased relative to the background. And interestingly, we also saw that for the Hodgkin-Huxley neuron. If you run a white noise analysis on the Hodgkin-Huxley neuron, you also see a structure very like this, which is a little bit unclear actually what that means in terms of the underlying biophysics. So there were Hodgkin-Huxley-like neurons, and there were also neurons like this that came out very nicely from this analysis. So that in this case, what you're seeing is that there are kind of two types of stimuli, right, that trigger spikes. And one way to understand that is that you can have in the retina on-off cells, neurons that care about the light turning on, and neurons they also care about the light turning off. So they care about a change in the stimulus independent of the sign. Right. So if you were to calculate the spike triggered average for those stimuli, there'll be some examples where the light went on, some examples where the light went off, and basically they kind of average to almost zero. But if you use the covariance analysis, that actually pulls out a space in which you can properly capture that bimodality, right? You have additional information that lets you now see that there are really two clouds of stimuli, the on features and the off features, and to be able to predict the behavior of the neuron in ways that calculating the spike triggered average just wouldn't do. So anytime you have sort of symmetries in the input, that means that the spike triggered average is not necessarily a good representation. So, a simple example is an on and off cell, as we just talked about. Another example would be a neuron that cares about, say, a little local fluctuation in the input, maybe a little brief period of a sine wave, but it doesn't really care about the phase of the sine wave. And so every time you get an example of that, you can shift that phase by a certain amount so that all the examples that you get then kind of average away to zero. And so the mean won't be very useful, but if you do the covariance analysis, you would find a basis in which you could capture that phase invariance. All right, so this idea of computing the covariance modes of the spike triggering stimuli is one way to get a richer model that is very helpful in cases like this. There are other ways of dealing with this spike history effect that other variants of building models like this that also have been used a lot in the literature. And one is one that directly adds in, right, this spike history influence. So we saw it sort of emerging. The importance of spike history we saw has a role in changing the kinds of features that you get if you look at the covariance structure of the inputs. But why not just try to model that directly and to build a statistical model that incorporates not just a stimulus filter, but also this post-spike filter. And so that's been a lot of work in the last decade or so has used methods like this. Some of you who are statisticians might recognize this as a generalized linear model. So generalized in the sense that there are linear filters that govern the relationship between input and output, but generalized in that it allows for this sort of nonlinear relationship between the linear projections and the output. And so here's an example where that really does just statistically formulate the model that Marcus Meister's lab generated. So now we try to predict the firing rate as a nonlinear function of the stimulus convolved with a stimulus feature and the firing rate response itself convolved with a feature. And so you also might recognize this as essentially an autoregressive model, right? So the output depends on itself. So it's regressed not just on the drive, on the external variable, but also on itself. Now, in order to fit models like this easily, one constraint that they have is that that one can fit them using optimization procedures if these nonlinearities are chosen to be they're fixed, right, and they're chosen to be in the exponential family. That is, they have the form of e to the something, right? So this nonlinearity is like exponential of this, for example. And so if you make that assumption, what you can do is write down the likelihood of your output, of your observations, your spike data, given the choice of the model, and then maximize over the parameters of the model. So what does likelihood mean? That means that's the probability of the data given parameters of the model. So it's thinking about the data's probability as a function, right, of the parameters of the model, and then you can crank through an optimization procedure and find the best values of the parameters of f and of h. So rather than do what we did, which was just directly to calculate the mean and the covariance of the data, here instead what you will do is in every time bin you'll count the number of spikes. That will be the firing rate. You need to include a noise model, and we assume that the neurons are noisy. And so, what's the probability of seeing k spikes in a given time bin of size t? We assume that comes from a Poisson distribution governed by the firing rate at time t. So you take the rate at time t, multiplied by the time bin. So this is just the form of the Poisson distribution. Take that to the power k. multiply that by the exponential, again, of the rate multiplied the time bin. So, you can look up the details of this, but under this assumption that says that all that matters for the firing of the neuron is the firing rate at a given time t, but it's a stochastic process, so that will generate, something between 0 and n spikes. Probability of seeing a certain number, k spikes in that bin, is governed by that time varying firing rate. Now, what's the time varying firing rate? That's generated by this model, right? There are some parameters to the nonlinearity. There are parameters to the filter shape and the shape of the autoregressive filter. Yeah, you set values for those. You write down this Poisson model. You take all of your data. You count the number of spikes in every bin k. You look at your input, right? You generate the firing rate. And now you keep varying the parameters of F and H until you find the parameters that maximize the probability of the data that you generated, or that you have from your system. And so that is a very powerful method. It always works, right? You always get you get an estimate of the filter, you get an estimate of the post-spike filter as well, which is certainly a advantage over these methods where you don't have an explicit description of what is happening internally to the neuron after a spike. There is a downside to it though, which is that you have to select in advance this nonlinearity. So in the methods that I talked about already, the nonlinearity just emerges from the data, right? You look at your spike triggering distribution, and here the nonlinear function is really just these blobs, right? It's built from the histogram of the spike triggering stimuli themselves. Whereas in this model, you just choose that in advance. You say it has a particular form, and all we're going to fit is sort of the slope, let's say, of the nonlinearity. Okay, so we spend a bit of time comparing different variants of how to fit these kinds of models, and I think coming back to one of the questions from earlier on, it works very nicely when you're controlling the stimulus from the outside. But that has all kinds of limitations. We can make a nice random white noise stimulus, but generally the real world isn't a white noise stimulus. So even not worrying about stimuli that are being generated from within, what if the animal itself is generating the stimulus that it's responding to? For example, if you're a rat, right, and you're whisking to explore the world, we know that neurons in somatosensory cortex care about the whisk movement, but the animal's generating that. So it does it in any way that it wants to. That stimulus is generally quite correlated and very structured. And you could never get it to do exactly the same thing twice. So if you want to test your model, generally what we would do when we build one of our models is we would do our experiment in the retinal ganglion cells many times, right? Like you saw in the very beginning, we had those rasters of retinal ganglion cells, and you saw that they responded very reliably. Really, we would like our model to be able to predict those reliable firing rates. If we have an animal that's behaving naturally, we can't get a raster, right? They will never whisk exactly the same way 10 times in a row. And so we have to think about different ways to characterize the model apart from just building PSTHs, of looking at the firing times and how reliable those are. And so here's some examples of a whisk, right? So now you can see what I was talking about already, that the whisk itself is very regular, right? They tend to fire at about to whisk at about 10 hertz. And here's a spike train from a neuron in barrel cortex that was firing in response to this animal whisking. And now you can see the problem, right? That how do you know, right, which model is better? And so here we've compared a bunch of different ways of predicting the times of these spikes, and the colored lines are the time varying predictions, and I've overlaid here the gray, the actual times of the spikes. And, you know, you can stare at that and you can see that these different models are doing okay, right? I mean, maybe there are moments when these probabilities of firing are high that kind of overlay with the spike times. There are other times when they're high, but there is no spike, but that could just be noise on that particular trial. How does one compare, right, between these different estimates of how the neuron is firing? You know, so here we compared spike triggered average, the covariance approach, where we now use multiple features, a couple of different variants of that higher dimensional model. The generalized linear model, where we just assume now one feature, but that the neuron, that we're also incorporated spike history. And also just a much more standard way of thinking about what's happening in these neurons, which is that they are phase tuned, that, you know, that the cell fires at a particular phase of the whisk, right? So that's a kind of more Hubel-Wiesel style, you know, identification of what seems to be a reasonable parameter, which is the phase of the whisk, and then just asking how well does that do as a coding model. So what is best right here? What is a good way to answer that? Well, remember that I just talked a bit about how one fits, right? The generalized linear model. And there what one does is to maximize the likelihood of the data given the model. So why don't we just do that for all of these models? So rather than asking how well we can reproduce a particular PSTH, let's ask how likely was the data under each of these models. And the GLM was specifically designed to maximize the log likelihood. And so, you know, in this case, it does a little bit better maybe, but you can see that one might have hoped that was sort of the answer, right? Look at the log likelihood, but generally we found that the log likelihood doesn't actually differentiate these models particularly well. There's a very narrow range of values of the log likelihood, and that it doesn't really help us too much. And so what we did instead was to compute the coherence between the predicted output and the spike train. Right, so the predicted output was that time varying probability, and we have the spike train. We can just take the Fourier transform of the prediction, Fourier transform of the spike train, and multiply them by one another. That gives the coherence, and that also gives us a value that's not just one number for the whole data set. Right, here is what we get with the log likelihood. Instead, we get to see how well the prediction matches the reality as a function of frequency. So it gives us a little bit more information. And so here the different models are all overlaid as a function of frequency. And so the blue is that tuning curve that just cares about the phase of the whisk. And you can see that does the worst in this case, right? The coherence here is very low. But the others, maybe as we should have realized from the log likelihood measure, they're all kind of similar. You know, there are examples, you know, there are some frequencies over which, you know, maybe the GLM does a tiny bit better than the others, but generally they're all more or less indistinguishable. There are other examples, however, like this one, where there's considerably more difference between them. And so here's a case where the GLM actually does quite a bit worse at low frequencies than some of the other models. You know, over some range of frequencies, they're all fairly equivalent, but the spike triggered average and the covariance analysis does considerably better for a wide range of frequencies than the GLM. And finally, here's a case where it turned out that the tuning curve approach worked better. And so here's the predictions from the phase tuning. You see it's almost perfect. And overall, the frequency ranges, that model actually outperforms all of the other examples. All right, so that is, I'll post that paper for you so you can read through the details of some of the methods that I've mentioned. I mean, there are many others as well. I've talked mainly about the case where you could use random stimuli to characterized coding. If I get time, I'll talk a little bit about this variant that is a bit more like a GLM model that's extended to multiple features. This sort of almost turns into a little bit of a neural network type model. One can also use neural networks, right, to fit the whole thing. You have input, you have output. You can just, use deep learning to predict the outputs, and that's been applied actually very successfully recently to neurons in the retina. I guess my slight quibble with that, compared to the things that we've talked about already, is that the models we've discussed already give you a feature, right? They tell you at least an approximation of what it is in the input that neuron is sensitive to. Whereas if you fit a neural network, you don't have that, right? You have millions of parameters in a black box that's potentially as complicated a thing to understand as your original system. And so it really depends what your goals are. If you're an engineer who's trying to precisely estimate what it was in the input that someone was watching while they were sleeping, right? If you want to build a BCI, you may want to just throw, as many parameters into your model as possible and build the best possible correspondence between input and output. If you're trying to understand the system and you want to know what it is, that's being extracted from the input, then you may want to go to simpler models that give you, a concrete estimate for what that filter is. Okay, so some of you raised some of the kind of caveats already. Let's just review them. So we talked already about the possibility, right, that there may be other stuff going on that you're not dealing with. The neurons within the network themselves are generating all kinds of complexity that can mask or make the relationship between input and response complicated. One would like to be able to understand this at the level of neural populations. And so, everything we talked about so far was really just focusing on how to predict the firing of a particular neuron. One can expand that to populations by building, a kind of GLM that applies to the entire network. So now every neuron has a GLM, but now we couple all the neurons together. So you take the output of neuron 1 and you feed that into neuron 2 as part of its drive. That is, while one might think about that as a population method, it's really not. What it does is to fit for a particular neuron in the presence of the firing of all the other neurons. So you're really thinking about the rest of the population as part of the stimulus. So that does a little bit get at the question of internal variables, right? There are ways to handle internal variables if you have access to their firing rates. If you can't see them, then there's nothing you can do about that. So ultimately, we know, right, that neural systems do have lots of stuff going on that is not being characterized typically in our experiments, and there's been a lot of really great recent work to take that into account. We have traditionally, when we're trying to characterize the visual system, we've given visual inputs and assumed that was the leading drive to those inputs. Recently, people have seen that visual cortex is full of information about movement, right? As the animal is sort of making nose movements or whisking, or its body is moving, a lot of correlated activity that is correlated with all of those movements. It also shows up in visual cortex, which can really influence, right, and affect the quality of the model that we're making, and really does make us kind of rethink, the goal, right? That really, particularly in the mouse, a lot of activity in cortex is this much more holistic picture of both, the incoming stimuli and the animal's own behavior. And so that tells us we have to keep track, right, of parameters like arousal and movement and potentially other sensory variables as well. We talked a bunch here about the retina, which mainly because it's all these methods work very well there. As one moves deeper into the visual system, the kinds of features that drive neurons get more and more complicated. You probably are familiar with convolutional neural networks that have been used to characterize visual systems, or at least that are a leading way of thinking about how the visual system might work. If one looks in intermediate layers of networks like that are trained end to end to predict or to be able to characterize images based on images, based on pixels, then you see things in intermediate layers that seem to resemble some of the properties of V4 and other kinds of higher order layers in the visual system. But these methods tend to not work very well because they have, as you move higher and higher into the visual system, you gain invariances, right? So the same image moved around over a range of space, right? All equally well drive the neuron. So if we were just to do a simple spectral average, then we'd just get a smear, right? And so those issues, right, the increasing invariance as one moves up through the visual system, make it increasingly hard to apply simple methods. And so that's where more interesting and more complicated statistical methods to characterize inputs are kind of needed. There's also all kinds of top-down effects, along with movement. There are more cognitive effects that also influence firing in sensory cortices. So in this case, mismatch signals is a wonderful paper from Georg Keller and Mark Hubener a few years ago. So what they did here was to have rodents running in a virtual reality environment. And so here recording from visual cortex, what they would do from time to time is just break the relationship between the movement, as the animal's running, the world is moving by them. And from time to time, they would just disconnect, right, the feedback. And so something would happen, the animal would move, but the visual world wouldn't respond in a way that it should have, right, based on the movement. And so that's kind of a feedback mismatch. And so you find that there are neurons, even in visual cortex, that care strongly, right, about those moments of feedback mismatch, so that seem to have an expectation of what the world should do and respond when that expectation is broken. So again, that makes the problem of figuring out coding more complex and more interesting perhaps. And just final example here, there are other influences that are kind of semantic. We know that visual cortex gets a huge amount of feedback back from higher order areas, and here is one of the things that does. So here, this is called the size illusion. If you, a ball or an object at the back of a scene like this appears larger than one at the front, because we're taking into account, the structure and the perspective of the scene. So influences like that, it's being demonstrated, show up directly in V1, right? So you would imagine that the size effect is something that we kind of process higher up, but it turns out that these two circles show up as different sizes, even in V1, which we would have thought just replicates what's happening on the retina, but in fact also already incorporates interpretation of the scene from the point of view of perspective. All right, I think I will take a momentary break, because we're about to take a switch into talking about adaptation. So let's just see if there are any questions.
Right, but I think, Vijay, are we going to do that now, or are we doing that tomorrow?
We're actually past the scheduled time, but Adrienne, do you have many more slides?
Oh, yes, I'm afraid I do, but that, why don't I I will really cut down what we're going to, what is the time? 7, gosh, yeah, sorry.
No, it's okay. I mean, I think one possibility is that, I don't know if you want to just show them. I know I think you're scheduled to also talk tomorrow, right? So I think you're going to
I could talk about these things tomorrow.
Well, no, but you could just very quickly, I don't know, put this in in some reading material or something, so students can maybe read it, and then in the very beginning, just give a very broad view. Just as a suggestion, Because I just don't know what the hard stop is for people.
Yeah, right.
And for you as well.
Yeah, that time went really fast. So yes.
We're
Okay.
asked a lot of questions, which is good.
Well, which was great, and I'm glad.
Yeah.
You know, I normally do this on the Blackboard, and it's just so much easier for me and for you too, because we can really go through the details. So I have really a lot I could tell you about adaptation, but let me try and just squeeze it into just a few words. So one of another issue, right, in characterizing coding, right? What do we mean by coding? We have some input, we have some output, and we want to make the map, right, between input and output. But neurons are dynamic, right? They have internal processes that don't just cause refractoriness. You know, we talked a bit about that, but they also cause sort of longer timescale changes in the internal nature, right, of the internal state of the neuron that can change, that can influence the way they respond over time. And here's, you know, just a very simple example that if you have a stimulus and you change it and you show another stimulus, you know, the firing rate has these time constants of response that such that, you know, what is the you know, the code here, right? If you take this stimulus, which of these values of the firing rate is the appropriate one, right, to say is the output for that stimulus. And so there, you know, there are all kinds of mechanisms for generating history dependence, ion channel dynamics, synaptic dynamics, network dynamics that give kind of short-term memory properties to the system. And so one of the roles of adaptation that we have explored quite a bit is to understand, you know, as the neuron is dealing with stimuli that change in their nature, how do the mappings from input to output change, or should they change, or do they change? Because very typically, as an animal is moving through the natural world, the statistics, you know, of natural inputs are hugely variable. So this is just a sound wave, right? And so you can see that the amplitude, right, of the sound or the stimulus, it could also be, you know, whisking as an animal is exploring the natural environment, is changing over kind of orders of magnitude. Right, so one would like, and the same thing is true for the visual world, right? As you look around the room, even you look from a window to the rest to you know, the interior as you drive from, you know, inside a car park to outside, the light level changes over, you know, 10 orders of magnitude. So how does our sensory system deal with these huge variations in amplitude? You know, if we think about our neuron, right? Remember our nonlinear tuning curve that, you know, that takes the stimulus, projects it onto this feature, and now its firing rate varies from, you know, 0 to 100 over some range of stimuli. How can that possibly encode things that are varying, right, over 10 orders of magnitude? And so what we have explored is, rather than thinking about this case where you just change the stimulus from one value to another, instead we change the stimulus from one distribution to another, right? So if, you know, how does a neuron encode fluctuations that are happening at some, you know, overall amplitude compared to how it would work if you change that overall amplitude? So if you just run a stimulus like this, different white noise input, and you switch it from one state to another, then you see that neurons adapt to that change in amplitude in the same way that they adapt to a change in stimulus, right? So there is some slow change in the firing rate of the neuron that is accommodating that change in amplitude. One would expect that neurons would change their coding properties, and there's a whole, you know, line of research going back to the 50s that predicts that should be the case. Sensory systems, in principle, should be trying to maximize the amount of information that they represent about the input, and their goal should be to make best use of their available firing rates, right? So let's imagine a neuron can respond, you know, let's say here from 0 to 10 hertz. What it should try to do is to spread that dynamic range, right? It should make those firing rates as meaningful as possible about about the strength of the input. And one analytical way to do that is to look at what the distribution of stimuli are in the real world and to parcel them up so that approximately the same number of stimuli, down in this range get mapped onto the 0 to 1 range as stimuli up here get mapped onto the 9 to 10 range, right? So we should stretch out the dynamic range of our input-output map so that we map out all of the range of inputs in the world. And that would maximize the amount of information that our code carries about the input. And so if we have this case, as we see in the real world, where the strength of the input is varying over time, what should an optimal neuron do? One thing it might want to do is to look at all the ranges of inputs that ever get generated over its lifetime, and then map those out, figure out their range, and then set your input-output curve to accommodate as best you can, right, all the possibilities. That's not what real neurons do. Instead, they track the size of the stimulus and then adjust their responses in real time. And so we showed that directly in the fly neuron. We take inputs that, we take a white noise signal that varies over amplitude, and we characterize the input-output mappings in different windows of time throughout that presentation. And if we plot out the input-output curves taken, evaluated in these different windows, then we find that they're not all just the same curve. In fact, they change, right? And they stretch their representation of the stimulus in a way such that it matches the overall scale of the input over those times. So if we normalize the stimulus by its amplitude in this window, then we see it's actually the same input-output curve, but where it's dynamically able, right, to stretch that range over time. So that's one of many aspects of adaptation that I could tell you about. Well, I'll just get through a bazillion slides here, and I just get to the end, which I wanted. I was way over optimistic here. So here, I've posted these slides so you can have a look through. There are many different ways that adaptation changes neural coding. So one, we see changes in the feature that's being extracted. We see changes in the nonlinearity, as I just showed you. You see spike frequency adaptation, where there are these slow responses to changes in the stimulus. And you also see something that's called like an oddball paradigm, where if you show the same stimulus many times, you get adaptation to that. You show a different one, you get an enhanced response to that novel stimulus. And there've been many theoretical ideas that have been put forward to explain these different aspects of adaptation, efficient coding, predictive coding, redundancy reduction, where one is trying to remove aspects of the stimulus that are predictable. You want to enhance the surprise of novelty. So all of these really deeply build in the idea that neural systems incorporate knowledge, right, of some aspect of the environment. That they're not simply black box input-output machines, but they are constantly tracking the behavior of the statistical properties of the world, right? And they are adjusting their own behavior in a way that takes into account those statistical properties. And so that is kind of the take-home message, right? That a lot of the questions that were asked about coding, I think, are really relevant to that, because what is all that internal structure doing? It is building up, presumably over time, right, as some kind of internal model of the world that then is influencing the way incoming information is processed. And so while we started out with a pretty naive, view that we could build a map from input to output, really what we should be thinking about is how knowledge of the world that is part of, our cognitive system is then feeding back to influence the way our brain is extracting meaningful information, perhaps in a way to optimally continue to build those models. And I think you're going to see examples of this in thinking about how flies navigate, right? How they use olfactory information to do a very task, goal-directed thing, which is to find the source of the input. You don't necessarily want to represent all the details, right, of an olfactory source. You want to just know where's it coming from. And so there are that sets what you want to pick out of the sensory input in a way that depends on the task that's trying to be solved and your knowledge of the environment. Okay, let me stop there and take any additional questions. Also happy to take them on Slack later if people have to take off.
Great. Well, thank you, Adrienne. So I don't know, I think the people, if you're willing to stay on a little bit, I think people can maybe ask some questions. I think, let me just put, I think there was one question that was posed a couple of times, and I'm not sure I fully understand it, but I think there's a question, I think, from Priyan saying, like, what is the tolerance level when you consider the dot product between the signal and the feature? So I don't know if you can parse that question and answer.
Yeah, I'm not completely sure what you mean. So
yeah, I don't know if Priyan wants to speak up. I don't know.
Yeah,
Let's
I think one way to think about that is that, you know, there is something, right, a feature in the input that is meaningful. One way of analyzing the stimulus is by filtering it through that feature, but that's obviously not precise. And so, you know, it's not that the neuron only cares exactly about that. There is some, you know, that's only ever an approximation. And where that tolerance, I guess, comes out in the wash is in the shape of the nonlinearity, right? Is in the shape of that input-output mapping, because it could be the case, right, that there is something super precise about the input that a particular neuron cares about, but maybe it's a much more complicated thing than just our one-dimensional filter. And so what that will look like is it will show up as a kind of a smoothing of the decision function. You know, so there's a kind of dialogue, let's say, between how precisely one's able to capture the feature dependence with the set of filters that we're using, and our estimate of the relationship between the stimulus and the firing rate. And so some of the failure of approximating the feature can kind of come out as a smoothing, let's say, of the decision function.
Great. So Vijay, I don't know if people should raise their, I guess, the reaction hand if they have a question. Yeah, because I don't know how to unmute people. Like, I can ask them to unmute, but I don't know if I have the. You may have to unmute people.
No. If people have questions, you can just raise their hands and we can unmute them. But
as Adrienne said, as Dr. Fairhall said, that she can also answer some questions on Slack if people want to do that.
Adrienne, there were a bunch of questions. We can also save the chat and email it to you. So you can also on Slack. That's also fine.
Yeah, let me see if there's anything. Oh yeah, was it reasoned out by the phase tuning method 1 in that case? Yeah, no, well, okay, there is, here's my theory, which is it actually relates to what we just saw in terms of adaptation. So if you're, you know, if you care about whisk dynamics, right, and you care about some feature, right, of the whisk dynamics, if you had this adaptation property constantly happening, then that would sort of normalize that feature, right? So if you are constantly scaling your input-output curve according to the recent past of the stimulus, then what that would do is actually get rid of the amplitude of the feature and turn it into a phase coder. So I think phase tuning could be a reflection of a neuron that is adapting to the variance, the local variance of the input. So that's at least my theory about why in that case for that neuron, you know, that might have been a neuron that was a good adapter.
Excellent. So I think, I don't know, I feel like, you know, we're kind of over the time and people may be kind of nervous. I think that if there people can think about this, look through your slides and ask questions there, we should also let you go. Thanks again for taking the time.
Okay.
So early in the morning for you.
Yeah, thanks. See you.
Yep. So Vijay, I don't know, you end the session whichever way.
Yeah. So one quick announcement. So tomorrow we resume at the same time, but remember there's a Python bootcamp organized for some of the people who have signed up. So the first session for the Python bootcamp starts tomorrow at 1400 hours Indian time. So people who have signed up, please do turn up at that. So we conclude for today then, and come back tomorrow. Take care, everyone. Stay safe.